Hello students, the topic of today's lecture is recombinant DNA technology. Earlier we have studied the tools of recombinant DNA technology. Today we will study the process of recombinant DNA technology or steps involved in recombinant DNA technology. Now what is recombinant DNA technology? Recombinant DNA technology is isolation of desired gene from an organism, its integration into a suitable vectors, and then introduction into another organism. It aims either to obtain multiple copies of the desired gene or the protein produced by this gene. Recombinant DNA technology is one of the remarkable advancements in biotechnology. This technique was developed by Boyer and Cohen in 1973. Processes of recombinant DNA technology. This is a general process of recombinant DNA technology, which involves several steps in specific sequences. These sequences are isolation of DNA, fragmentation of DNA by restriction endonucleases, isolation of desired DNA fragment, ligation of the DNA fragment into a vector, transferring the recombinant DNA into the host, culturing the host cell in a medium at large scale, and extraction of the desired product. Now we see step by step the process of recombinant DNA technology. The first step is isolation or extraction of DNA. The first step in recombinant DNA technology is to isolate the desired DNA in its pure form, that is, free from the other macromolecules. Since DNA exists within the cell membrane along with other macromolecules such as RNA, polysaccharides, proteins, and lipids, it must be separated and purified, which involves enzymes such as lysozyme, cellulose in plant cells, chitinase in fungi, ribonuclease which degraded RNA, and proteases which degrade the proteins, etc. Other macromolecules are removable with other enzymes treatment. The addition of ethanol causes the DNA to precipitate out as fine threads, and purified DNA is isolated. Second, restriction enzyme digestion. Restriction enzymes act as molecular scissors that cut DNA at specific locations. These processes are called restriction enzyme digestion. They involve in the incubation of purified DNA with the selected restriction enzyme at conditions optimal for that specific enzyme. These restriction enzymes are also called molecular scissors. The technique Agarose Gel Electrophoresis reveals the progress of the restriction enzyme digestion. This technique involves running out the DNA on an agarose gel. The negatively charged DNA travels to the positive electrode and is separated out on the basis of size. This allows separating and cutting out the digested DNA fragments. The vector DNA is also processed using the same procedure. Now come to amplification using PCR. That once we get the desired DNA, we can amplify it by using polymerase chain reaction and then ligate with the cut vector directly. For PCR, the reactions are run on thermal cyclers and it requires the component template, the DNA to be amplified, primers, all chemically synthesized oligonucleotides that are complementary to a region of the DNA polymerase and Taq polymerase, nucleotides needed to extend the primers by the enzyme. The PCR helps to amplify a single copy or a few copies of DNA into thousands to millions of copies. This technique is advancement over the cloning vector. The other way of amplification of the desired DNA is cloning. The molecular cloning is the laboratory process used to create recombinant DNA. There are two fundamental differences between the methods. One is that molecular cloning involves replication of the DNA within a living cell, while PCR replicates DNA in the test tube, which is free of living cells. The other difference is that cloning involves cutting and pasting DNA sequences, while PCR amplifies by copying an existing sequence. These are the diagrams to show how PCR works. Next step is the insertion of the desired gene segment in a vector. The desired DNA is inserted into a suitable vector, which may be plasmid, bacteriophage, or cosmid. Vector is also cleaved by restriction enzyme and may result in sticky or blunt end. The choice of vector is mainly based on choice of host and length of DNA inserted to be cloned. Plasmid has cloning capacity of 5 to 10 kb DNA insert. Viruses have 10 to 25 kb DNA and cosmid have 40 to 48 kb DNA. The DNA fragment is inserted into the vector and ends are sealed by means of DNA ligase. DNA ligase acts as a glue. The result of this step is recombinant vector. These are the diagram which shows how recombinant DNA is formed. You can see here the plasmid. Plasmid is found in E. coli, which is extrachromosomal double-stranded circular DNA, which is autonomous body and replicate itself. The desired gene is isolated from the human cell. It may be plant cell or animal cells. Here, growth hormone, example of growth hormone have been shown. The desired gene of for human growth hormone is isolated. Meanwhile, the plasmid is also isolated from the bacterial cells. Both the DNA cleaved by restriction endonuclease EcoR1, then the desired DNA is joined into plasmid vector. There is a DNA recombination process, and finally a hybrid DNA is found. Now this plasmid with desired DNA, the product is called recombinant DNA or chimeric DNA. This DNA, recombinant DNA, is reintroduced or reinserted in the bacterial cell for containing gene for human growth hormone. These are the whole process of cloning into a plasmid. There is introduction of recombinant vector into a host. The host bacterial cells are mixed up and are left culture in medium having polyethylene glycol or calcium ions. The recombinant plasmid gets multiplied once it is introduced into the E. coli cell. With each division of E. coli, plasmid also multiplies. The DNA segments are then isolated and used for various purposes. There are two types of objective. First, expression of cloned gene for production of protein, and second, introduction of cloned gene into the host plant or animal so as to give transgenic plant or animal. The whole process is known as cloning. Cloning is nothing but it is a just like a Xerox. Next process is insertion of recombinant DNA into host. For this, recombinant plasmid and bacterial cells are mixed up and are left for culture in medium having polyethylene glycol or calcium ion. In this step, the recombinant DNA is introduced into a recipient host cell, mostly a bacterial cell. This process is called transformation. What is transformation? The entry of recombinant DNA into the host cell is called transformation. Recombinant plasmid gets multiplied once it is introduced into the E. coli cell. With each division of E. coli, plasmid also multiplies. The DNA segments are then isolated and used for various purposes. Next point is isolation of recombinant cells. The transformation process generates a mixed population of transformed and non-transformed host cells. The selection process involves filtering the transformed host cells only. For isolation of recombinant cell from non-recombinant cell, marker gene of plasmic vector is employed. We can understand it by example. PBR322 plasmid vector contains different marker gene. PBR322 contains two antibiotic resistant gene, that is ampicillin resistant gene and tetracycline resistant gene. When PST1 restriction endonuclease is used, it knocks out ampicillin resistant gene from the plasmid so that the recombinant cell becomes sensitive to ampicillin, and we get transformed cells. Now the recombinant DNA or desired DNA is stored in gene bank or gene library. Now, what are the application of recombinant DNA technology? Recombinant DNA is widely used in biotechnology, medicine and research. Recombinant DNA is used to identify, map and sequence genes and to determine their function. Recombinant proteins are widely used as reagent in laboratory experiments and to generate antibody probes for examining protein synthesis within cells and organisms. Many additional applications of recombinant DNA are found in industry, food production, human and veterinary medicine, agriculture, and bioengineering. DNA technology is also used to detect the presence of HIV in a person. Recombinant DNA technology is also used in gene therapy. In gene therapy, we can correct the gene defects which give rise to hereditary disease. Application of medicines. Insulin production by DNA recombinant technology is a classic example. Recombinant DNA technology is also used in clinical diagnosis. ELISA is one of the examples which involves recombinant DNA technology. Recombinant DNA technology is also applied in agriculture. BT cotton is one of the example of recombinant DNA technology which is used to protect the plant against bollworms. After getting the recombinant DNA, through recombinant DNA technology, which is also called genetic engineering, in this, unrelated organisms can be brought together. Such genes are called transgenes, and organisms having foreign genes are known as transgenic. For production of transgenic plants, agrobacterium plasmids, that is TI and RI, which are respectively found in Agrobacterium tumefaciens and Agrobacterium rhizogenes. Both these plasmids have T region which become integrated into host genome. Here in this diagram we can see how desired DNA is inserted into the TI plasmid. The same process, the plasmid, TI plasmid and is isolated, cleaved by restriction enzyme, and the desired DNA is integrated into the TI plasmid. Then this recombinant DNA plasmid is inserted into plant, which is the another process is called gene transfer in plants, which is done by the another vector called gene transfer vector, that is agrobacterium. And when the new tDNA carrying new gene is inserted into plant, the plant expresses with new trait. So how this is the desired character we can get into another plant which we want to express.